All right. Very provocative, Sean. So we'll come back to that in the discussion. So I'm going to take a few minutes to describe to you a program that we're developing in ASCO to try to learn something from the off-label prescribing of targeted therapies for patients with advanced cancer who have a genomic test performed. So we've been talking all day today about whether it's worthwhile or not to do genomic profiling on patients with advanced cancer, and what do you do with the results once you get them. Of course, this is something that can and is happening every day right now. Patients with advanced cancer are getting genomic tests performed in their cancer centers from commercial laboratories, et cetera. Doctors are struggling with how to make decisions about what treatments to use. And importantly, the outcomes of those patients are not being captured in any way that we as a community can learn from them. So we are missing a lot of information that might be informative. So we're developing a program that we've now named the Targeted Agent and Molecular Profiling Registry, or the TAPER study. So this is the problem. I just stated it. Patients with advanced cancer who've exhausted all standard treatment options, they have some sort of a multiplex genomic profiling test performed. There's a potentially actionable variant that's detected. The clinician faces an immediate problem of how to get the drug. And we heard from Lillian, Sue, and others about the low proportion of patients for whom a drug match is actually available. And there are a variety of reasons for that, but one is simply the fact that, there may be drugs out there that would be appropriate, but because they're not standard of care therapies in that particular clinical context, they may be inaccessible to the patient for one reason or another, oftentimes related to insurance coverage. And perhaps most importantly, if the patient gets the treatment and gets access to the drug, receives the treatment, how do we learn from the experience of that patient in receiving that treatment? So you've seen charts like this all day long, and I'm not going to belabor this, but the point being that we're getting all this molecular subsetting done. There's all kinds of drugs that are potentially out there that align with these various mutations that are being detected in a whole range of common tumors. And what are the potential sources of drugs for a doctor and a patient? So, of course, you have commercial drug use within indication. That's really not what we're talking about here, because that's the standard of care. You have commercial drug that could be used off-label. That's perfectly legal to prescribe that, but it may be limited by reimbursement. Of course, there's always the option for patients to participate in clinical trials, but we know that trials are not always available, patients are not always eligible, patients sometimes choose not to participate, and the overall rate of clinical trial participation in this country remains at a low level. And then in certain circumstances, usually after a drug has finished its initial testing, when there's an NDA under review, the sponsor of the drug may decide to set up an expanded access program, but that's a voluntary activity, and many sponsors choose not to do that, but it remains an option. The program that we're setting up focuses on the use of commercial drugs prescribed off-label. So we're not talking about investigational drugs. I want to say that about 3 different times. And I think what you'll see and what I'm going to describe is that in many ways, what we're setting up could be viewed as the off-label companion to the NCI match study. It has many features in common. So the sort of overarching goals are to facilitate patient access to marketed, targeted drugs against common aberrations, to create a registry that captures the administered treatment and patient outcomes. And of course, to do this requires participation from all the key players, the patients, of course, physicians, pharma companies, the payers, and perhaps the FDA. The way we envision this working, and this is not up and running yet, but it's in development, we are hopeful that we'll get the pharma companies to actually provide the drugs at no cost to the patients. The patients would have to agree to data collection. They'd have to consent to participate in an IRB-approved protocol that will operate this registry. The physician will have to submit the required follow-up data as specified in the protocol. we hope the payers will reimburse for the routine clinical care costs of caring for patients, particularly because they won't be asked to pay for these expensive drugs if we can have the drug companies provide the drugs at no cost. And then a third party, which in the future would be ASCO under the auspices of our rapid learning healthcare system, CancerLink, that's currently in development. At the moment, it might be an independent third party, would host the outcomes registry. So it would probably look something like this. A genomic profiling test would be performed using an analytically validated assay in a CLIA certified laboratory. The patient would be enrolled in the registry. The physician would submit the genomic profiling data and a proposal to use one of the drugs that's available through the program. Here we refer to an honest broker. This is likely going to take the form of a molecular tumor board. I'll come back to that in a minute. Would review the physician's proposed treatment plan against predetermined guidelines specified in the protocol and would approve the use of the particular drug that the physician has selected from among those available in the protocol, or could alternatively make a different recommendation for the treatment of the patient. And I'll come back to that point as well. The patient would then receive the drug treatment for a pre-specified period of time. Their outcomes would be captured in the registry. The cost of their care would be reimbursed. And at the end of the day, all of the participants in this program would share in the data that's collected in the registry. So we are now in the process of developing this into a formal protocol. The primary objectives are shown here to describe the anti-tumor activity and toxicity of commercially available targeted anti-cancer drugs used off-label for treatment of patients with advanced solid tumors with a known genomic variant. We may include some hematological malignancies in there as well. We've already had some outreach from the Multiple Myeloma Research Foundation that has expressed a high interest in collaborating with us in this. And then a sort of a co-primary endpoint to facilitate patient access to these commercially available targeted drugs prescribed off-label. And then a variety of secondary objectives to record the treatment-related adverse events, to create a prospective registry of patient outcomes, to create a prospective registry of the tests that are used. And I'll elaborate on this point in a minute, but the specific genomic profiling done in this proposal would be done outside of the context of the protocol. In other words, the doctor, and we're trying to set this up to mirror to the greatest extent possible clinical practice as it currently is going on. So the doctor would have to make a determination in discussion with the patient that doing some sort of genomic profiling test is clinically reasonable at that point in the patient's care. The doctor would have the discretion to order whatever test is available to them in their sort of commercial marketplace. They could use their hospital laboratory, they could use a cancer center laboratory in their region, they could send it to a commercial laboratory. Whatever they would ordinarily do, they do. The active enrollment of the patient in the protocol doesn't really begin until the doctor gets the profiling results back and determines that there's a variant that's detected that is potentially targeted by one of the drugs that is available through the protocol. So the eligibility criteria would be the following. Patients with advanced solid tumors, as I said, possibly myeloma, for whom no standard treatment options exist. They have to have adequate organ function. We would allow performance data 0 to 2. We have to have some eligibility criteria, but we'd like them to be broader than the traditional eligibility criteria used in many cancer treatment trials. And then we've had a lot of discussion with a lot of stakeholders. And this, in part, was the reason I asked the question this morning of the panel members about tell me how to define a quality test, about whether we should allow certain tests or any tests to qualify a patient to participate in this study. We've determined that the best we can do right now to ensure that a test is of reasonable quality is that we will allow tests which is a genomic test, so either a FISH, PCR, NGS, that's been performed in a CLIA-certified, CAP-accredited lab that has obtained a McKesson Z code. I'll come back to that in a minute. And if the lab is located in or offering services in New York State, it must also have obtained New York State accreditation. As far as we can tell, those are the standards that would have to be met these days to define a high-quality test being performed in a high-quality laboratory. The issue about the McKesson Z code, so some of you may be aware of this or not, but McKesson Biosciences, in an effort to try to learn about the nature of molecular tests that are actually being performed and to make some sense out of them, has set up a system called the McKesson Molecular Diagnostics Exchange. And what it essentially does is allow a laboratory to register its test on the Molecular Diagnostics Exchange. And in doing so, it has to describe what the test is, how it's performed, what its intended use is, and some basic performance characteristics of the test. And McKesson then puts this through an internal review process, and if they determine that the test is, quote, unique, then they assign a Z code identifier. And you can go right now to the McKesson Molecular Diagnostics Exchange website, and you can find the whole list of tests that have these Z codes. One of the advantages of including this is that it would allow us to capture some basic information about the nature of the test that's being performed, so that potentially we can use this as a vehicle to learn something about the test itself. I'm not going to take you through this schema for this study. It's a bit complicated, but I did want to just point out that there is up in here, so let's see, over here, a molecular tumor board. And the way we envision this working is that based upon the drugs that are made available for the program, we will include in the protocol a list of the available drugs and a list of the known targets of those drugs based upon the FDA-approved label for the drug and other information available in the literature. And the clinician will get the genomic profiling results, look at those results, determine if there's a variant in the patient's test report that matches to a variant that's on the list that's associated with the drugs. And if so, might choose to select the drug that hits that variant. And if there's, in a sense, a perfect match that's described in the protocol, then the treatment can just begin, assuming the patient meets all the eligibility criteria for the study. But there'll be many cases where there will not be a perfect match, or the data will be incomplete, or there'll be variations in what's detected in the test from what's listed in the protocol, or the doctor, based on knowledge, experience, whatever, may believe that another drug listed in the protocol, other than one that seems to match to the genomic profile of the patient, might be the preferred treatment. In any of those kinds of circumstances, the doctor would be required to submit the case for review to a molecular tumor board that will convene to support this protocol. And then the molecular tumor board will evaluate the case and make a final determination as to what the best options might be for the patient. So why the molecular tumor board? Frankly, we think that this will protect patients from inappropriate treatment based on incorrect interpretation of molecular test results. We've had some concerns, and we've heard throughout the day about how complicated these tests are, how variable the reports are in terms of the supporting information that's provided to the clinicians. We're not confident that the doctors will necessarily be able to interpret the test results accurately and reliably. So we want to be sure that the molecular tumor board can assist in that. Similarly, it's not necessarily a given that the doctors will actually know how all the drugs work in terms of what their molecular pharmacology is and whether or not the drug is actually appropriate for the variant that's detected in the profile. So we want to protect patients from inappropriate treatment based on misunderstanding of the drug action. If we're going to do this, we're interested in comparing what the physician selects as the treatment option and what the molecular tumor board recommends as the treatment option. And then we've heard some pushback from some of the pharma companies expressing concern about their being accused of off-label promotion of their drugs, which of course is illegal, and the notion that this could offer some protection to them from any such allegations. There will need to be a set of rules that the molecular tumor board will have to work from in order to adjudicate the proposed match. This is just an example of what such a rule set could look like, but it's a levels of evidence approach, as we heard discussed earlier today, surrounding both the level of evidence that the variant is biologically consequential and potentially clinically actionable, and also a level of evidence approach that the drug is appropriate for the variant of interest. What are the potential actions of the molecular tumor board? Well, they could concur with the physician plan. They could recommend a treatment with another drug that's in the protocol that targets the selected variant. They could recommend treatment with a drug that's in the protocol but targeting another variant that's present in the patient's tumor. They could recommend off-label treatment with a drug that's not in the protocol. So they may determine that, maybe the most actionable variant that's detected in the patient's tumor is best treated with a different drug that doesn't happen to be available through the protocol but is otherwise commercially available and could still be prescribed. Or they could recommend patient participation in a clinical trial of a novel agent that seems to be an appropriate match to the genomic profile of the patient's tumor. This is very much a hypothesis-generating study, if you will. We're not going to prove anything from this. We're looking for signals. We're looking for signals, that are based, in a sense, in the real-world assessment of patients to the extent that we can, make this look as close to the real world as possible. So we're going to use objective response rate as the primary endpoint. This is a non-randomized, uncontrolled study. We can't use a time to event endpoint and expect to get any reliable results, so we'll use objective response rate. We'll collect data on progression-free survival, overall survival, duration of time on treatment, perhaps as a surrogate or estimate of whether the treatment is being effective or not. Of course, we'll collect high-grade adverse events and serious adverse events. The statistical analysis plan defines each tumor type, variant, drug match as a group. So you could think about bladder cancer with a BRAF V600E mutation treated with a BRAF inhibitor. That would be a group. The plan is to enroll 8 patients per group. If we see no responses in the first 8 patients, we would stop enrollment and conclude that there's not a sufficient signal of activity to continue. If there's at least 1 response in the first 8 patients, we'll plan to enroll an additional 16 patients. If we see 4 or fewer responses in this group of 24, we'll declare insufficient interest. And if there are 5 or more responses, then we would believe that we have a signal of activity that's worth pursuing further. Regarding the data collection, the plan is to collect patient demographics necessary to confirm eligibility, of course, to collect the genomic test that's performed and its results, the proposed treatment plan, the molecular tumor board recommendation, the most recent prior treatment that the patient received and the patient's best response to that treatment, and then the efficacy and safety endpoints. If we're able to set this up, who potentially benefits? Well, we think that a study like this could generate benefit for all of the stakeholder groups. Patients would receive the targeted agent that's matched to their molecular profile. Physicians would receive assistance in interpretation of the molecular test results, guidance and treatment recommendations, and access to drugs. The pharma companies that participate would receive data on drug use and outcomes that could inform their research and development plans and lifecycle management of their products. Payers would also receive outcome data on drug and test use, and which could be useful to informing their future coverage decisions, and the same for the regulatory authorities. So this is my last slide. You know, there's a lot of discussion about this going on now in a lot of venues. Those of you who are attending the Friends of Cancer Research Brookings Conference on Clinical Cancer Research in a couple of weeks will hear this discussed again on a panel at that meeting. And you know, the key issues really center beyond the operational issues, which are formidable. You know, like if we don't get any drug companies to participate, we don't have a study. But putting that aside, you know, some of the key issues that maybe we'll get to in the discussion today are, will the data be reliable, generated through a mechanism like this, and how might it be used? You know, would the data be used for hypothesis generation to inform new studies? Could it be used for label modification, for example, new safety issues that surface in a population of patients that didn't participate in the original clinical trials of the drug? Could it be used ever for label expansion? You know, could the data be sufficiently compelling that it could actually be used to support a new indication? Could the data be robust enough to support compendia or guideline modifications? We've heard a lot about that today. Could it be used to support reimbursement policy, to expand or potentially reduce coverage? Because it's, you know, highly probable that in many cases we're going to find that drugs are not effective in certain genomic matches. And finally, you know, how will data like this be useful in informing doctor and patient decision making? So we'll come back to those issues later on, but for now, thank you. To round out this session, we wanted to ask someone who had a lot of experience working with registries to give us a reality check as to the challenges of utilizing databases and registries to actually generate information that people would find interesting and reliable. So very pleased to have Garnet Anderson here from the Fred Hutchinson Cancer Research Center to talk about some of those issues.